In this module, we'll be discussing cancer and the process of carcinogenesis and how cancer cells come to be. Before we really get into that, though, you need to kind of understand how normal cells work. And then you can understand how cancer cells come to be a little bit more deeply. So what we're looking at here is the normal cellular growth process of what's called differentiation. So all of our normal cells that we can see in our body, right, they all grew up what they wanted, grew up into what they were supposed to be when they grew up, right? So they start as a stem cell. They start where a single cell, a normal human cell, starts as a stem cell in the beginning of creation, and then it turns from one type into two types and then into several other types, and it differentiates into the cells that they're going to be when they are fully grown, mature cells. So in other words, a multipotential hematopoietic stem cell can turn into platelets, red blood cells, mast cells, which are a type of white blood cell that release histamine for inflammation, myeloblasts, basophils, here are all the white blood cells. Here's all our lymphocytes, another type of white blood cells involved in specific immunity. This is just an example of how our normal cells differentiate into what they want to be when they grow up. Once they are fully differentiated, they are fully functional, they're doing their job, they're doing something good and functional for the human body. Once we understand how normal cells work, we can look at the regrettable process of undifferentiation. So this isn't a stem cell turning into a fully functional cell. These are fully functional cells turning into non-functional cells, something that does not aid the body, something that doesn't have a purpose, doesn't have a function. They just undifferentiate from something that has purpose further and further away from purpose until all they do is cause disease. That is the process of grading cancer. We're not talking about staging or how far spread cancer is, but grading. And what grading is you look underneath microscopes or in a pathological biopsy to look at cells to see, are they normal? Are they nearly normal? Or are they going down in the gradient to becoming few normal cells and mostly just abnormal cells, meaning they are going through a process of change. It's called cellular adaptation, where they become less and less like their parent cells, and they continue to replicate further and further away from those fully differentiated cells and become what's called completely undifferentiated. So the undifferentiated cells, as far down as possible, these are completely abnormal and completely undifferentiated. These are what are referred to as aplastic cells. Aplastic cancer cells.